Hello everyone and welcome to Instant Biology by Dr. Neela. I am with you here again with another very important topic, that is mechanism of action of enzymes. Now before I start this topic, I would want to tell you that I have already taught this topic in Hindi. So if you are more comfortable in Hindi, then you can watch that particular lecture. The link to that lecture would be given in the description section also, and it would be available at the end of the lecture on the top. Now, before we start this particular topic, let me just brief you what we would be studying in this topic today. In this topic, I would be telling you that how enzymes function, what are the parameters on which the action of enzymes is visible, on which parameters enzyme do not show any effect, what do you mean by binding energy, what is transitional state, and also I would be talking about the graph that is more or less represented when the enzyme activity is talked about. And I would also be talking about the delta G value with respect to the enzyme activity, and we would be comparing the enzyme catalyzed reactions and non-catalyzed reactions also in this particular lecture. After this, we would also be dealing with some of the PYQs that have been asked in different life science examinations. So without wasting much time, let us talk about the mechanism of action of enzymes. So if you see your screen, let me just first of all scroll to the first slide. How do enzymes operate? Now, as I said, when I was describing the topic briefly, I told you I would be talking about the basic of enzyme action. Now, first thing or the first point that has been mentioned over here is that the enzymes enhance rate of reaction of for about 10 to the power 7 to 10 to the power 9 times. What does this mean? This simply means that if I am using a particular enzyme, then the enzyme would increase the speed of the reaction. Now you would always be knowing this, and this has been this is taught in very early classes also, that what do enzymes do? Enzymes increase the rate of the reaction or increase the speed of the reaction. Now, by what factor does the enzyme increase the speed of the reaction? It is roughly around 10 to the power 7 to 10 to the power 9 times. Now, let me explain it to you in another way. Let us say that the speed of the reaction is x. Okay. If the speed of the reaction is x, then when you are using the enzyme, then the speed would be now becoming 10 to the power 7x or 10 to the power 9x. That means the speed would be increased by a factor of 10 to the power 7 to 10 to the power 9. Coming to the next point, the enzyme lowers the activation energy. Now, what do you mean by activation energy? I would be talking about that in just a moment. But for now, you just have to keep this in mind, that how does enzyme increase the rate of the reaction? It increases the rate just by lowering the activation energy. I would be discussing about activation energy in just a moment. Next important thing is that it does not change free energies of substrate and product. What do I mean by that? Now, for example, the free energy of substrate is delta G substrate, delta Gs, and the free energy of the product is delta Gp. Now, these were the free energies before we were using any enzyme. That means it was the free energy of an uncatalyzed reaction. But now, when you are using the enzyme for the catalysis, then also the delta G for the substrate or delta G of the product will be not modified or will not be changed. Next, this is the same thing that I have written in the previous point. That means delta G remains unchanged, and since delta G is remaining unchanged, therefore the concentration or the amount of the product and the reactant at equilibrium remain unchanged. What does this mean? Now suppose I have reached the equilibrium of the reaction, or the equilibrium of the reaction has been reached, then the concentration of the product over here and the concentration of the reactant, whatever concentration of product or whatever concentration of reactant was present at the equilibrium, it will not be changed by the enzyme activity. The concentrations will remain the same. So what we have understood up till now, delta G would not be modified. The concentration of the product and the enzyme at equilibrium will not be modified. Now you will be asking me that Sir, if nothing is being modified by the use of the enzyme, then why we are using the enzyme? The main important thing why we are using the enzyme is just because it is increasing the speed of the reaction. What do I mean by this? Let me explain it to you with a real life example. Now suppose I am living in Lucknow and I want to travel to Delhi. Delhi is approximately 500 kilometers from Lucknow, and there are different ways in which I can reach Delhi. For example, I can take my car, I can go by bus, I can go by a train, and if I am in a hurry, I might opt for flight also. Now, all of these things, all of these vehicles, they will be allowing me to go from Lucknow to Delhi. That means my initial position and the final position will remain the same. But the only difference is that if I am using different vehicles, they will allow me to travel at different speeds. That means I am reaching Delhi, no problems in that. That means the final product is reached. But what is the time required in which I am reaching that? So, in the same way, when I am using an enzyme, the equilibrium is reached in a very short time. That means if you are using a non-catalyzed reaction, suppose there is a reaction in which you are not using any enzyme, then it will require a lot of time to reach the equilibrium, right? But when you are using an enzyme, comparatively lesser amount of time would be required to reach the equilibrium. That is why we use the enzyme. So suppose let us say in an uncatalyzed reaction, let us say 2 hours were required for reaching the equilibrium. When you are using the enzyme, let us say only 5 minutes were required for reaching the equilibrium. So this is the difference that enzyme creates. It speeds up the reaction, it decreases the time required for reaching to the equilibrium, and this is extremely important thing in biological reactions specifically. Since we are talking about biochemistry, so I am more inclined towards talking about biological reactions. Moving on to the next slide. Enzyme accelerates the attainment but does not shift their positions. Now what do I want to say over here? The position of the substrate, or the free energy of the substrate, rather than position, I must write over here that free energy, that means delta G of substrate and delta G of product, they will remain the same. That means the free energy that is present inside the product or the free energy that is present inside the substrate will remain the same. However, the enzymes will, or the enzyme will hasten, enzyme will increase, or it will accelerate the speed at which the equilibrium is obtained, or the reaction speeds up. Now, let us talk about how does enzyme operate, or how does enzyme function. Understand this, that let us say that this is the structure of an enzyme. Okay. Now, enzyme has a particular site which is called as, or which is referred to as, active site. Now suppose this is the enzyme and this site over here is referred to as active site. Okay. Now in order to explain it to you, I have drawn it over here. Now this is the active site of the enzyme. You can see the marking over here. This is the active site of the enzyme. Now remember, active site comprises of two individual sites. That means the active site is comprising of two different regions. One region is called as binding site and the other region is called as catalytic site. Binding site, as the name suggests, the substrate would be binding at this particular site, and catalytic site is the site where this substrate will be getting converted into the product. So this is simple and you can understand it by the name itself. The next point says that specific substrates can bind at the binding site. Now what do I mean when I say specific substrates? This is a very important point. Enzymes are very specific in their functionality. That means if an enzyme has substrate A that binds to its active site, substrate B will not be able to bind to its active site. Why am I saying that? Because the active site of an enzyme has specific functional groups. Those will be allowing the activity with only the substrate A and not with substrate B. So substrate B will not be able to fit inside the active site of the enzyme only, or even if it fits, it will not be able to interact with the functional groups present in the active site. Okay, so for example, let me just draw it over here, that this is the enzyme, and let us say this is substrate A and this is substrate B. So primarily substrate A will be able to fit in over here, but substrate B might not be able to fit in. That is why I'm saying that the enzymes are very specific in their functions. Let us come to the next point. The interaction between the active site of the enzyme and the substrate is always weak non-covalent interactions. Now, when I say weak non-covalent interactions, what do you understand from this statement? Weak non-covalent simply means those bindings or those bonds that are not very strong. For example, hydrogen bonds. For example, van der Waal interactions. For example, hydrophobic interactions. So these interactions are not very strong in nature. Why only weak and non-covalent? Why am I saying non-covalent? Because covalent interactions are very strong in nature. So all the interactions that will be present over here will be non-covalent in nature. Why am I talking about weak interactions only and not strong interactions? Understand this, that as I said, that let us say this is the enzyme and this is the active site over here. Now, if the interaction between the substrate and the active site is through strong bonds or through covalent bonds, now you would know that in order to break these covalent bonds, a lot of energy is required. As I've said that these are strong bonds, therefore in order to break the strong bonds, a lot of energy will be required, and it becomes almost impossible to break these strong bonds. Therefore, if at all there are strong bonds that are present between the substrate and the active site of the enzyme, the substrate will be permanently locked onto the active site, and this enzyme will now be non-functional because no other substrate is able to bind over here. This substrate has been permanently fixed. Therefore, this enzyme will become non-functional. Enzymes are very costly. That means in order to form an enzyme, it is a very costly affair. Therefore, the body system does not want that the enzymes becomes, waste. They are of no use. The system does not want that. Therefore, non-covalent interactions, weak interactions are always involved in the interaction between the substrate and the active site of an enzyme. So I hope all of these points are clear in your mind. Now let us move on to the next slide. In the next slide, we can talk about the binding energy. Now, what do you understand from binding energy? Let us quickly discuss this. Binding energy is the free energy that is released in the formation of, let me just highlight important points, free energy released in the formation of large number of weak interactions between the enzyme and the substrate. Let me just draw the structure of an enzyme over here again. Let us say this is the enzyme. Okay, now what I'm trying to draw is I'm trying to draw a substrate over here like this. So this is a substrate. Now what happens is when the substrate enters inside the active site, what happens is the weak non-covalent interactions, something like this, are formed between the active site and the substrate molecule. Now, because of these weak interactions, what happens is free energy is released in the formation of large number of weak interactions between enzyme and substrate. When the substrate is binded to the enzyme, what happens is weak interactions are formed between the active site of the enzyme and the substrate, and formation of these weak interaction leads to a release of large amount of free energy, and this free energy is referred to as delta G. Now, this free energy has a very important role in the functionality of an enzyme. I will be telling about the connection between this free energy, the released free energy, and the activity of the enzyme in just a moment. But you have to understand what is binding energy. Binding energy simply means the amount of energy that is released when the substrate forms weak interactions between the active site of the enzyme. So this is clear. I believe you would have got a clear idea. Now, there were two models basically that were used in order to explain the activity of an enzyme rather. So the first model was lock and key model, and the second model was induced fit model. So first of all, let me just discuss about the lock and key model. Lock and key model was given by Emil Fischer in 1894. And what does it say? It says that it assumes that there is a high degree of complementarity between the shape of substrate and the geometry of the binding site of the enzyme. Lock and key model, as the name suggests, it says that the enzyme functions as a lock and the substrate molecule functions as a key. Now, as there is a fixed key for every lock, as you would be knowing that, right? No other keys are fit inside a particular lock. There is a fixed key for every lock. So in the same way, the lock and key model allows or says that the structure of the lock would be in perfect complementarity with the key. One key only will be fitting inside this lock. So let me just quickly show you the diagrammatic representation that I want to talk about. Over here you can see that there is an enzyme. This is an enzyme. Now this enzyme has an active site over here, right? So this is the active site. Now this over here is also the active site, and this over here is also the active site, right? Now understand this, that the structure of the substrate over here is in perfect complementarity with the structure of the active site. That means the active site, the substrate will be completely fitting inside the active site. As you can see that this is a square structure and this is a triangular structure. They will be completely fitting inside the groove of the active site like this. So what happens is the enzyme substrate complex is formed, and this leads to the formation of enzyme product complex, and finally the products are released from the enzyme. This is called as a lock and key hypothesis. So if I want to explain it in one sentence, it simply means that in lock and key model, the enzyme and the substrate will be in perfect complementarity with each other, and the structures, both of their structures, will not be changing, right? So this is about lock and key hypothesis, right? So now let us move on to the next model that was given in order to explain the activity of the enzyme. The next model that was given was induced fit model, as I've already said. The name of the scientist that gave this model was Daniel Koshland in 1958. And he said that the enzymes are flexible in nature. What did he say? He said that the enzymes are flexible in nature. What do you mean by flexible? It simply means that the enzymes can modify their structure. Depending upon the structure of the substrate, the enzyme can modify its structure. This is in stark contrast with what we have studied in lock and key hypothesis, because in lock and key hypothesis we said that the structure of the enzyme and the substrate were fixed, but over here we are saying that the enzyme can do modifications in its structure depending upon the structure of the substrate. So shape of an active site can be markedly modified by the binding of the substrate. This is what is induced fit model. Let me just show it in the diagrammatic representation. Over here you can see the structure of the enzyme, and this is the active site. Now you can see that this over here is a square, and if you just check out the structure of the active site, it is basically like a triangular shape. But as the substrate is coming close to the enzyme, as you can see, has modified its structure, and it is now able to incorporate the substrate. So enzyme substrate complex is formed over here, and then the enzyme product complex is formed, and finally the products are released out and the enzyme is now left. This is what is called as induced fit model. I hope you would have got a clear understanding. Now let us move on to the next point. What do you understand from transition state theory and activation energy? Firstly, let us talk about what is a transition state. Now, transition state is an intermediary state of the reaction. Now, we know that the substrates want to get converted into the product. Now, the substrate cannot directly get converted into a product. There is a transition state in between. Okay. Once the substrate reaches to this transition state, and the transition state is the state that is present at the highest energy. So those substrate molecules that have this much of energy, they reach to the transition state, and then only they can form the product. Okay, so it is an intermediary stage. Additionally, it is an unstable state. Unstable state. What do I mean when I say this is an unstable state? I mean that the substrate cannot be present over here for a longer period of time. They have to get converted into product. And if they are not able to get converted into product, they will be reverted back to the substrate. Right? So transition state is the state that is present at the highest energy, highest delta G value, and only those substrate molecules will be able to reach this transition state, those that have this much of energy. And once they reach the transition state, only then they can form the product. Otherwise, the product will not be formed. Okay, so at this particular state, the molecules are neither at the substrate level or neither at the product level. It is somewhere in the middle. So this is what is meant by transition state. Next, transition state is the point of highest free energy, as I've already told you. That means the delta G value over here will be highest, in which substrates partially get converted into products. Okay. Now, what do you mean by activation energy? The next point that I want to discuss over here is activation energy. Activation energy, as the name suggests, it is the difference in the free energy between the transition state and the ground state. I'll be showing it to you with the help of a diagram, and then you would be able to understand it in a much better way. But for now, just understand that the free energy, the difference in the free energy between the transition state and the ground state. That means, suppose the transition state is over here, the ground state is over here. The difference between the transition state and the ground state is called as the activation energy. Now, Gibbs free, and it is also called as Gibbs free energy of activation, and it is also referred to as delta G, and the symbol that is used is delta G, something like this. This is called as activation energy. Now understand this, I'll be showing it to you that if the activation energy of a particular reaction is very high, what do you mean by this? When I say activation energy of a reaction is very high, I simply want you to understand this, that the difference between the delta G value, the difference between the delta G of transition state and the difference and the ground state is very high. This difference is very high. When the activation energy is very high, that means this difference is extremely high, the rate of the reaction will be very, very low. So there are different reactions that are running inside the biological system. Some reactions have a very high activation energy. Some reactions have a low activation energy. When I say high activation energy, you should understand that this reaction will be going at a very slow pace. Why I want to say that? Because if the difference between the transition state energy level and the ground state energy level is very high, then it is ought to happen that the number of substrate molecules that will be crossing this energy barrier or the transition state barrier would be extremely low. For instance, if I say we are starting with 100 substrate molecules, let us say only 2 or 3 would have that much of energy to cross the activation energy barrier. Understood? Did you understand my point? Now, if I want to say that the activation energy of a particular reaction is low, this means that more number of substrate molecules will be taking part in the reaction, and they will be having more number of substrate molecules will be having that much of energy to cross the activation energy barrier. So more number of substrate molecules will be getting converted into products. This will lead to a speedy reaction. That means the reaction will be extremely fast. I would be, whatever I just said, I would be talking about that using the help with the help of a diagrammatic representation also, so that you can get a better understanding about the same. Now let us move on to the diagrammatic representation. Okay, so this is what I was talking about. Let us understand this from the beginning. On the y-axis over here, you can see the Gibbs free energy, Gibbs energy, or you can also call it as Gibbs free energy. And on the x-axis, there is a reaction coordinate. Now, on the top over here, this is the energy that is called as transition state, or this state is called as transition state. Okay, and this over here is called as ground state. And I've already said that the difference between the energy of transition state and ground state is called as activation energy. So in this particular case, in the A case, you can see that the activation energy is pretty large. The substrate or the reactant molecules have this much of energy in the beginning. The reactant molecules or the substrate molecules have this much of energy in the beginning. Now, if I consider this A, let us say 100 molecules of substrate were taking part in the reaction. Obviously, since the activation energy is very high, this much is the energy. So the substrate molecules that will be having this much of energy will be very, very less. So let us say only 2 molecules out of 100 were having this much of substrate energy. So only they will be getting converted into the product. This will imply that the rate of the reaction will be extremely slow. Now, on the contrary, let us say that we are using an enzyme over here. So this was the uncatalyzed reaction. Uncatalyzed. That means over here no enzyme was used. But let us talk about this reaction. Let us say that this is catalyzed reaction, the B1. Now you can see that this is the transition state over here and this is the ground state over here. Obviously, the energy difference between the transition state and the ground state has decreased significantly, and this is called as activation energy. On comparing the two, that means the A diagram and B diagram, you would obviously understand that the activation energy in B has significantly reduced. Now, if we consider in the same way that let us say 100 molecules of substrate were present, because the activation energy has lowered down, of course the substrate molecules that will be taking part in the reaction would be, let us say, 50, because the activation energy has lowered down. So since the 50 molecules are taking part in the reaction, they are getting converted into the product. Obviously the speed of the reaction will increase, the rate of the reaction increases. That is why we are constantly saying that enzyme increases the rate of the reaction. In other words, we can also say that the enzymes provide an alternative pathway for the reaction to proceed, because this was one pathway and the enzyme provided another pathway, another way in order to complete the reaction. So this is what exactly happens, and this is how exactly the enzyme does or perform their function. Now, this is another diagram. We are going to talk about this also, but this is essentially saying the same thing. It's just that the catalyzed as well as the uncatalyzed reactions have been shown in the same diagram. So over here you can see that, let me just show it to you. So this is the activation energy of uncatalyzed reaction. That means in which the enzyme is absent. And over here you can see the presence of enzyme. That means the enzyme is present. So you can see that the activation energy of the reaction has decreased significantly in the presence of enzyme as compared to in the absence of enzyme. So the same thing is being talked about over here, and that is why the rate of the reaction increases significantly. Having said this, we have completed what I wanted to tell you about the functionality of the enzyme or mechanism of enzyme. Now I want you to understand or to solve some of the PYQs that have been asked in CSIR, GATE, DBT, etc. examination. Let us start. So the first question over here is, which of the following models best explains substrate induced conformational changes in enzymes? Now obviously I told you, when the substrate will be inducing any change in the enzymatic structure, then this type of this model that was given was induced fit model. Remember, one another model was lock and key model, but in lock and key model we have already said that the structures of both the substrate and the enzyme remains fixed, they are not changed. Now let us move on to the next question. Which of the following statement is true about transition state? So we are talking about transition state over here during the enzyme catalyzed reaction. So transition state has lower free energy than the substrate. No, transition state has the highest free energy. Next, enzyme binds transition state more tightly than the substrate. True. Enzyme will be binding the transition state because enzyme will be binding the substrate very tightly at this particular point, because after this point only the enzyme will be getting converted into product. Something like this: enzyme plus substrate. Enzyme substrate complex has been formed in the transition state, and finally enzyme plus product will be released. Okay, next point is transition state is more stable. No, it is unstable state. Enzyme increases the free energy of the transition state. No, the free energy of the transition state is decreased. Only then the rate of the reaction increases significantly. So the correct option over here is B. Let us talk about the next question. Enzymes increase the rate of biochemical reactions by, we have talked about this multiple times. Increases the activation energy. No, activation energy is decreased, decreasing the overall free energy change, stabilizing the transition state, and next altering the equilibrium constant. So decreasing the overall free energy change is the correct answer, because the free energy is decreased, the activation energy is decreased. Activation energy is what? It is the difference in the free energy of between the transition state and between the ground state. So that is significantly decreased, and then only the rate of the biochemical reaction is increased. So correct option is B. Now, which one of the following is not affected by enzyme activity? So activation energy is affected by the enzyme activity. Yes. Reaction rate, yes, it is also affected. Equilibrium position and transition state stabilization, this is also affected by the enzyme activity. But which one is not affected? Equilibrium position, that means the equilibrium constant, is not affected by the enzyme activity. Remember that the time in which the equilibrium is reached, that is of course affected, because the rate of the reaction is speeding up. So the time at which the equilibrium will be reaching, that would be significantly decreased, but the equilibrium constant or equilibrium position, it will not be changed at all. Next, what role does binding energy play in enzyme catalysis? So it destabilizes the enzyme substrate complex. It is used to overcome entropy and orient substrate. It increases activation energy. It breaks covalent bonds in the product. Now the correct option is B. With this question, I want you to understand what is the role of binding energy. That is why I have put this question. Now the binding energy that I just explained a little while ago was the energy that was released when the substrate formed different weak bonds between the active site and the substrate, right? So when these bonds were formed, a lot of free energy was released. This we had seen. Now this free energy or this binding energy that was released is actually used to decrease the activation energy. Now you might wonder that why did the activation energy decrease primarily? You can see that the activation energy has decreased. Previously the activation energy was this much, and now the activation energy is this much after using enzyme. How was it possible? Because the first law of thermodynamics states that the energy can neither be created nor destroyed. How come the energy is decreasing over here? The energy is decreasing because this is compensated by the binding energy only. So when the binding energy is released, it is used up in lowering down the activation energy. And when the activation energy lowers down, the rate of the reaction increases. So in other terms, in other words, what we can say is it is used to overcome the entropy. Basically, it is used to overcome the randomness, because you might be knowing that when the randomness of the universe basically goes on increasing, but when the enzyme and substrate are binding together, the enzyme substrate complex has comparatively lower energy, lower entropy, and this entropy is lowered down by the binding energy only, and it is also used for orienting the substrate, or for changing the structure of the substrate, or for properly aligning the substrate into the active site. So that is why correct option over here is B. So I hope you liked today's lecture, and much of your doubts would have been cleared related to enzyme functionality. If you like the entire video, just hit the like button and do not forget to subscribe our channel. Also, I would want you to tell me which more topics you want to study. So do not forget to type it down in the comment section. Thank you so much and have a good day.